Hello and welcome everyone to Season 4 of Contact's Produce Safety webinar series. This series is part of a larger industry outreach and risk mitigation project called Contact, or Scientific Challenges in Cost-Effective Management of Risks Associated with the Implementation of Produce Safety Regulations. This project is supported by a specialty crops research initiative from the USDA National Institute of Food and Agriculture. Any opinions, findings, conclusions, or recommendations expressed in this presentation are those of the speakers and do not necessarily reflect the view of the U.S. Department of Agriculture. So many of you always ask, who is Contact? This is a huge grant that's representative of many universities and government institutions. It's comprised of more than 50 faculty, staff, students, and volunteers, not to mention the support of our Produce Advisory Committee, or PAC. It takes a village, and you're part of it. We're very appreciative of each individual's support and ongoing efforts to make Contact a grant championed by you all. Contact is now in its final year, and so we are very grateful for your support. As we're preparing for Contact 2.0, if there are things you would like us to focus on, questions you would answered, or resources you would developed, or if you want to write a letter of support or serve on our Produce Advisory Committee and help direct the content of this research and extension, please reach out to myself, Laura Strawn, or our fearless leader, Dr. Michelle Daniluk. I would also like to take a moment to highlight each and all of you. In our 4 webinar series so far, we've had over 4,400 unique viewers representing over 55 countries. I thank each and every one of you. And so with that, why we're all here today. Here we're going to hear from Dr. Linda Harris and Dr. Joy Waite-Cusic, Unpeeling the Science, Onions, Food Safety, and the Fight Against Foodborne Pathogens. And with that, I will turn it over to our extremely talented set of speakers.
All right. Thanks, Laura. So I'm Joy Waite-Cusic. I'm going to jump in, and Linda and I are going to go back and forth a little bit with our presentations. I'm Joy Waite-Cusic, Associate Professor of Food Safety Systems at Oregon State University, and I'm also a co-PI for the Western Regional Center to Enhance Food Safety. And then Linda is illustrious, and I'm sure everyone knows her, but she's Professor of Cooperative Extension in Microbial Food Safety at the University of California at Davis and is co-PI for the Western Center for Food Safety. So today we're going to talk through several different topics related to onions. I'm going to kick off talking about whole onions. We'll talk about a couple of the outbreaks, standard growing practices, some field trial work that Linda and I have both been involved in. We'll both talk about some cross-contamination research we've done and dry sanitation. And then Linda will talk mostly about fresh cut onions, outbreaks associated with those, handling practices, and pathogen behavior. So we'll go ahead and jump right in here. So just a reminder about kind of some of the food safety concerns that have kicked off a lot of the recent research. In 2020, we had a giant outbreak of Salmonella Newport that was epidemiologically linked to red onions grown in California. And we had over 1,600 cases associated with that outbreak. And then in 2021, we had a similar outbreak at around the same time. This was Salmonella Oranienburg, and also associated with red onions, this time onions that were imported from Mexico. And again, over 1,000 confirmed cases. These onions were a little bit different in that they weren't field cured. We'll talk about that in a bit. But both of these outbreaks were very large in terms of the number of confirmed cases, and 2 of the largest outbreaks we've had in over a decade for Salmonella. We want to review some of the background information that was associated with these outbreaks just because it did guide a lot of the research that Linda and I have both been involved in since that time. FDA and CFDA did publish outbreak investigation report. And so as part of that investigation, they did travel to the farms that were associated with the 2020 outbreak and collected a number of onion samples and environmental samples. And just to highlight those findings, there were over 1,100 onions that were sampled. None of those tested positive for Salmonella, which is not all that uncommon in outbreak investigations. But there were a number of environmental samples that were collected as well. So over 800 environmental samples were collected and analyzed. 26 of those were positive for Salmonella. 11 were positive for Salmonella Newport, so the same serotype associated with the outbreak, but none of them were the actual outbreak strain. So without that sort of piece of evidence that would put a bow on this outbreak, we're left with formulating some theories. And so in FDA's report, they provided us with at least their thoughts on the sources of the outbreaks. And none of these are all that surprising. So pointing out that a leading hypothesis was the use of contaminated irrigation water. Also, some concerns about adjacent and nearby land use practices, particularly the presence of livestock. And then there were also some plausible hypotheses, including bringing up irrigation water again. There was some sheep grazing on adjacent land, animal intrusion, large flocks of birds. And then in the post-harvest environment or in the packing house conditions, there were concerns about signs of animal and pest intrusion and food contact surfaces that had not been inspected, maintained, or cleaned as frequently as necessary to protect against cross-contamination. We're going to touch on the topics particularly related to water use pre-harvest and then packing house conditions later in the presentation. So these outbreaks were also fairly curious in that within each outbreak, the clinical cases, the isolates from the patients, were very closely related. So we saw very few mutations in these strains. And that helped to guide some of the hypotheses that we were generating. Our question was really about how do you get a population that is this similar? And our working assumptions were that there was one particular strain, a different one in each outbreak, that likely is growing to a very high cell density somewhere in the environment, potentially all on its own in a single location. And then that is distributed into the crop, and a lot of onions end up becoming contaminated. And then potentially that salmonella is going to survive or potentially grow during storage and distribution. And so this is a baseline hypothesis that we're working from. We believe that mass contamination, so the number of onions that had to be contaminated that led to this outbreak, is likely a function of the application of contaminated water. So this was, again, our assumptions going into the design of some of the studies that we'll present today. So for this next portion, I'm really going to focus on our efforts related to water applications and onion production and whether these could potentially explain salmonella outbreaks. So we'll talk through how water is used during onion production. Specifically, we're going to talk about storage or dry bulb onions, and then a little bit about whether there's any uniqueness to red onions. Just to get us all on the same page, here are all the things you might be calling onions. But for this presentation, we are not going to be talking about green onions or shallots or sweet onions. We're specifically going to be talking about dry bulb or storage onions. And over 80% of the U.S. crop is grown in Oregon, Washington, Idaho, and California. I'll introduce some differences between how we do things in the Northwest versus how they're done in California. So for Oregon, Washington, and Idaho, we pretty much exclusively grow what are called long day varieties, so meaning those that need lots of sunlight. In March or April, we seed the ground, and it's typically planted somewhere between 160,000 to 200,000 seeds per acre. In May and August, May through August, we have vegetative growth of those onion plants. And then in August, at some point, those onions start to go dormant. They're done growing. And so the tops will fall over, and we'll move those onions into a process of lifting them, breaking the roots, basically lifting them to the surface, field curing, and then harvesting those. And we'll talk a little more detail on that in a minute. And then from September through April, those onions are in bulk storage and then are distributed out into wholesale and retail markets. The onion production in California is a bit more diverse. So California has a lot of north to south differences in growing region and sunlight. And so in the far north of California, they will grow long day onions, very similar to what we do here in Oregon, Washington, and Idaho. And then in the Central Valley and the high desert, South and Central Coast, we see intermediate day and some short day onion production. And then in the far south, we have the short day onion production. And these are just some minor differences, but it also affects which onions end up going basically into the fresh market at different times of the year. For these dry bulb onions, we have 3 key stages that we were concerned about in terms of relative risks of salmonella and the behavior of salmonella in this particular crop. As we're nearing the end of that growing season, we have bulb maturation. So the bulbs are bulking up and getting ready to go into that dormancy phase. And then the plant starts to shut down. And so we would call this tops down. And this will signify when the grower will stop irrigating. And then we'll move into this field curing period. Typically in the Northwest, that period is several weeks, and we targeted a lot of our studies around a 1-month period. The field curing is sometimes shortened in order to hit that early market gap and to get a premium price. And that kind of depends on when the other short day varieties of onions run out. So there can be this gap in the market where there can be some improved value if you can get your onions to market sooner. Sweet onions, just to clarify, these are non-storage onions and these are not field cured. So our questions for our first field trials were really about whether this extended period in the field would mitigate risks associated with the use of poor water quality. We've conducted several studies to look at risks related to different types of water applications. I'm going to quickly go through drip irrigation because I think we can put a bow on that pretty quickly. Our first questions were related to drip irrigation. We did a study in Oregon. It was a greenhouse study looking at actually flood irrigation, but as a mimic for how drip might interface with the plant. And for this study, it was key that we compared a potential surrogate. In this case, it was the rifampicin-resistant E. coli cocktail to salmonella in order to be able to justify the use of that surrogate once we went to field trials. We did conduct field trials after that, and we looked at drip irrigation with different levels of contamination in Ontario Treasure Valley growing region. Linda also led studies in California looking at drip irrigation with contaminated water, again, using the E. coli cocktail. And then we conducted a recent study in Washington State, also looking at drip irrigation at 2 different contamination levels. So I just want to put a bow on this. All these field trials and everything have been very consistent. And I would like to say as simple as possible, we don't think drip irrigation is a problem. So we have field trials in 3 different states over several different years. At reasonable to extreme contamination levels, we see rapid die-off on the onions, and we don't seem to think this is going to be an issue at all. On the left, just to show the data a little bit, this is just an example of the data set from the Washington field trial. So on the left, we have the number of onions that tested positive for E. coli on different days throughout field curing. So in 2022, that's the darker gray, we had 13% of those onions tested positive on day 1. That was the only day in the whole study where we had any positive detection. On the right, in the yellow violin plot, you can see that the levels of contamination with E. coli were quite low. So we ended up with a maximum of about 20 cells per onion as the contamination rate in that case. That field trial was done at a moderate, I'm going to say a reasonable level of water contamination for this growing region. And then in the second year, we increased that contamination level just to force the issue related to drip irrigation concerns. So we increased the contamination level 100-fold to 100,000 CFU per 100 mil or 3 log CFU per mil, depending on which units you like. And the lighter gray shows that increase in contamination level certainly led to an increase in the number of onions that tested positive. We had a maximum of 68% after 1 day. And then that died off rapidly with curing. At the very end of this study, we did have 1 onion that tested positive at 2 cells per onion. Okay, what about other water uses? So there are a number of potential overhead water applications that could happen during onion production. And so we've done a couple of studies to investigate this as well. So Linda had done the drip irrigation, and then she had also done what we call the mock foliar spray application. And I'll show you some more pictures in detail of that. In Washington, with the drip irrigation side by side, we compared that to an overhead irrigation application. And then in Oregon, we also looked at the use of pesticide or clay applications if those preps had been prepared with contaminated water. So we wanted to look at that potential risk. And then we also followed up with another greenhouse study that I'll introduce a little bit of that data. We're still working on that analysis. In Washington State, this was the overview of our overhead irrigation trial. Again, this was compared with drip irrigation, at least the first year. So we had one variety of onions we were looking at in 2022. That was a yellow variety called Calibra. In 2023, we were able to expand that to look at 3 additional varieties. Ovation is a variety we had used in other field trials, and then we added a red variety and a white variety, both of which are commonly grown, at least in the Pacific Northwest. Our irrigation target was on the high end, so we wanted to apply as much contaminated water as might be reasonable in those growing regions, 0.4 acre inches, and our contamination level was 1,000 mpm per 100 mil. This irrigation level, and I'll start a video, led to approximate delivery of about 60 milliliters per onion of contaminated water. Droplet size is quite big for overhead irrigation, and this is our mock system. And so a lot of that water ends up running off of the plant. We collected samples immediately after that irrigation event, and then at day 1 in both years, and then you can see 2, 7, 15, and 28. Some of those were there were less sampling days in 2023 just due to resources. So what did we find? So in 2020, I'm going to show the 2023 data in this case, but we'll start with the yellow. So immediately after that overhead irrigation, as you might expect, we have lots of water contact with the plant. And so 100% of those onions did test positive for our rifampicin-resistant E. coli. The maximum cell density on those onions was 44 NPN, most probable number, or cells per onion is a way to think about it. As we went through curing, we saw a fairly rapid decline in the number of onions that were testing positive. And at the end of that curing period, we had no detectable E. coli in this case. That was identical to the previous year. So 2022 and 2023 were the same. We expanded again to other varieties. So we looked at white, the Cometa variety of onion. In this case, we had one onion that was quite a bit higher, but still these are pretty low levels. So 767 cells per onion. And we actually detected that on day 1. And then as we go through curing, again, the number of onions that test positive are rapidly declining. But in this case, we did have 3%, which I believe is 2 onions in this case, that tested positive at very low levels of contamination. Similar thing with red wing, so almost identical results. Again, a few onions testing positive at the end of field curing. So overall, curing is reducing risk in the case of this field trial. When we switch over to pesticide and clay applications, we had some other questions about whether this would be any different. So here's an example of how the pesticide applicator was applying either Pristine, which is our actual pesticide, or Surround, which is a clay application. So we had, in this case, 4 plots that went through each treatment. So 4 plots of the red variety that were treated with clay, 4 additional plots treated with pesticide, and then that repeated for the white onions. This pesticide application only led to a volume basically being distributed to each plant of about 2 to 4 milliliters per onion. So very little water ends up being applied. So let's look at this data. So same arrangement as before. So we're really looking at the number of onions that tested positive throughout curing. So in this case, we are applying very little water, and in a lot of cases, the leaf portion of the plant is preventing that water from contacting the actual bulb. So there's some shielding going on. So in this case, we're going to look at the clay application first. So the white onions, immediately after that application, we have about 55% that are testing positive. For the red onions, slightly higher at 63%. The number of cells is very low, so 12 or 17 NPN per onion in this case. After day 1, we had no detection of E. coli throughout the rest of the curing period in this case. When we switch over to pesticide, very similar. We did have slightly higher rate of initial bulb contamination. And we had one red onion at the end of curing that did test positive, again, at very low levels. So just 2 cells per onion in that case. When we switch over to 2023, we did basically as identical of a replicate as possible over these 2 years. And we're seeing honestly very similar levels of prevalence and pretty similar levels of contamination. We did have a few onions, again, testing positive at very low levels at the end of curing, 3% in this case, which I believe is 2 onions, and just at 2 cells. Pesticide, same thing. So we had very similar overall die-off during curing, but you will see there's a little bit longer survival during early curing in 2023 compared to 2022. So we did want to look at the differences in weather this particular year. So the top graph, the green is showing the temperature range per day, and then the black is showing specific humidity, and then the blue is showing the amount of precipitation on any given day. So 2022, the top one is very typical for the growing regions in Idaho and Oregon and Washington. There's basically no precipitation at all during the later part of the season. So it's very dry, very hot. In 2023, we had an anomaly. So we had a really significant rain event during the curing period. Actually, that was the day that we collected samples. So if you look over at the left, it's day 7. My poor students were drenched, but we did see some increased survival that we think potentially is related to that weather event. Now I'll jump into the California study. So far, we've shown a lot of risk mitigation associated with field curing, and we're going to see something a little bit different with the California study. So for Linda's study, they did a mock foliar spray, and you can see an example of that in the picture on the right. So their backpack sprayer, it's inoculated with, there's water inoculated with E. coli in there, and then each plant received basically a spray of approximately 30 milliliters. The contamination levels on these were substantially higher. So we had 2 levels, so 5 log CFU per mil in 2014, and then that contamination level was reduced in 2015 to 3 log CFU per mil. This is still 100-fold higher than what we did in the field trials I already presented. Let's look at Linda's data. So first, on the far left, you'll see the contamination level of the onions immediately after that contamination event. So we're seeing between 2 and 3 log CFU per onion. And then as we just go throughout the day, they took samples after 2 hours and 4 hours of that contamination event. And we're seeing a pretty rapid die-off. We saw some similar things with the Oregon study. But as that time was extended, some interesting things started to happen. So as soon as 8 hours, there was one onion that was showing elevated levels of E. coli. And that is, they went through 1 day, 4 days, and 7 days of curing. We're getting this huge amount of spread in the E. coli contamination level in this trial. As field curing was extended, and you can ignore the topped and untopped, that's just 2 treatments of the leaves. But in this case, we're seeing some onions are dying off. So we're not seeing any surviving E. coli in a portion of the onions. But then there's a substantial amount of the onions that are stable. The E. coli seems to just be out there. And then we have this troublesome section where E. coli, the evidence would point to it growing in this situation. This is the cause for concern, right? We want to know more about what's going on here. So we're still scratching our heads about that a little bit. Now I'll talk just briefly about the greenhouse study that we did as follow-up, mostly related to that result from Linda. So we wanted to see if we could produce a similar thing if we grew these onions in the greenhouse and applied a similar type of contamination. So we did a backpack sprayer, just like the field trial. We also wanted to ask some questions about if you applied water earlier in the season, would that contribute some differences to contamination rates? And could potentially that water get internalized into the bulb? And would that be an actual riskier situation? So we looked at different water applications, particularly related to pesticides that happen during the growing season of onions. And you can see just in the table, these are planted in March through May. We have some sort of pesticide application, say in April and in May and in June and again in June and later in June and in July and then very late in the season in August. So we had some questions about this. We did go ahead and design this greenhouse study to look at those, particularly related to survival growth and potential internalization in the onion bulbs. So we had 3 varieties of onions we grew here. We inoculated one set about 75 days or so prior to when we thought the bulbs would be mature. Waited a couple weeks, did another set, waited another week, couple weeks, did another set, and then we waited till they were mature and did that final set of contamination. And then we cured those in the greenhouse. So we sampled each set on inoculation day, at the last day of irrigation, and then at the end of curing. So I'm just going to show a portion of this data just to stimulate some thought here. For this particular set, this is going to be one variety that I'll present. This is just our yellow one. And once we figured out when they were mature, we back calculated when we had added or done the contaminated irrigation event. So that was 67 days prior to the end of maturity, 42 days prior to the end of maturity, 28 days, and then 0 days. So in this case, we analyzed 5 onions on the day of the contaminated irrigation event. We analyzed the leaves separately from the bulbs, and then we also blended the bulbs and did an enumeration that way. So the dots line up to the same onion in a row, basically. You can see if we just look at 67 days, we have 5 onions that were analyzed. Of those 5, 4 of the leaves tested positive. All 5 bulbs tested positive, and then 2 of the bulb blends also tested positive. And then the color code on the right is the level of contamination. So in this case, this is like immediately after we added the water. We would expect it to be about what the water was contaminated with, which was between 0 and 2 log mpm per sample. That's what we got. So same thing happened for that very last irrigation at the time of maturity. We also analyzed all the previously inoculated plants at that time of maturity. And so in this case, we can see that for 67 days, we had no positives. So 67 days ago, it was irrigated with contaminated water. We have no positive onions at that point. 42 days prior, we have 2 positive leaf samples, but no positive onions. A month prior, we have a leaf sample positive, and then we have 2 onions testing positive on rinse and 3 testing positive on blend, but they're pretty low. And then if we look at at the end of curing for all of these, we have no onions that are testing positive if they were treated with contaminated water way early in the season, midway through the season. No, 1 testing very low levels of positive at 28 days prior. For the ones that were contaminated right at the end of maturity, so what we would typically think is the riskiest situation, we had 2 onions test positive at the end of curing, and we went ahead and analyzed those by blending. And what was troubling about that is one of those onions was very positive for E. coli on blending. So we had 8 log CFU per mil in that blended sample. So we were definitely seeing, okay, most of the time we're seeing die-off. In certain situations, we may see growth. So what was going on? So Through all of our recent studies, we've taken efforts to photograph all of our onions and to cross-section and photo all of our onions, which is ridiculous. I have more pictures of onions than my children on my phone. But I want to show you what some of these onions look like. These are the 3 that did have higher levels of E. coli at the end of that curing period. And we had also dyed the water using a blue color so that we could see how the water was moving in the plant. So that top one is the one that's the most contaminated. And in this case, it also suffered from significant rot. So that potential release of sugar into the onion is probably contributing here. We can also see some blue internalization around the neck in the second onion, and then the purple one's hard to see because it's purple. Just to follow that up really quick, we did do some post-harvest analysis of one of our field trials, and we found that one of our onions that had slightly elevated level at the end of storage did also have some rot in it. I'm going to just caveat that we took thousands of cross-sectional photos of our onions, and this internal decay was only detected in this one onion. So we had a very healthy crop. It may or may not mean anything, right? So this is not statistically significant by any means, but we are interested in that potential issue of plant pathogen behavior and human pathogen behavior. Okay, I'm going to kick it over to Linda to talk about post-harvest storage and handling.
Okay. Thank you. All right. So I'm going to transition a little bit here. So we've got our onions curing in the field. And I know somebody asked about sacks. These things can be mechanically harvested, typically are mechanically harvested. But you can see in this one photo, I've got some additional curing going on in bins in the field. In this case, we've got some sacks of onions, which are sometimes used. So there can be a broad range of activities, but ultimately they end up at a packing house. And so at the packing house, this is where we're looking at a number of activities. It would be typical of a packing house: inspection, grading, sizing, and then ultimately in different forms of packaging at the end. So we wanted to look at the potential for cross-contamination risk factors in the packing house environment. So a couple of things at the broad level to think about. This is not a packing house where water is typically introduced intentionally. As was mentioned, a lot of this is grown and harvested in very dry environments, and so rainfall is not common during harvest. But it could happen, and so there could be unintentional or periodic introduction of water. But typically, these are dry environments. A lot of different types of contact surfaces, a lot of different types of surface material, and then a lot of opportunity for transfer modes. Onions to surface, surface to onion. And you can see there are sorters, human sorters, so there's also glove transfer and other types of transfer that are possible. When looking at this problem, we were thinking that we would have a contaminated onion coming into the system at some point and then contaminating that onion to surface. So you've got a contaminated surface. And we wanted to ask the question, how long do bacteria survive on surfaces that would be common in a packing house? And then also we wanted to understand what factors might influence bacterial transfer in these systems. So for the experimental design, we looked at typical conveyor belt material, polyurethane. So we obtained cut up conveyor belts, new conveyor belts. So we did not do this with used material, and then stainless steel. We looked at spot inoculation, drying on the surfaces, polyurethane or stainless steel, air drying, and then we put it into storage at
about 20 degrees Celsius, and then a relatively low relative humidity, which would be, again, relatively typical of the growing storage conditions in the California, Oregon kind of scenario. We used Ephesium, which is an organism that is often used as a surrogate in thermal processing. We wanted to know if this organism might be an appropriate surrogate for transfer studies. And so we started with this looking at the survival, and the Ephesium is in the solid square. You can see that we're looking at in the red here is the result at 12 weeks of storage, 84 days. And you can see two things happening here. One is that the curve, especially for Ephesium, is very different between polyurethane and the stainless steel. And the second thing is that Ephesium survives better than Salmonella in both of these cases. And so the survival is about a log difference between the two organisms at 84 days. So the other thing we did was to ask the question, if you've got onions running through these systems, you're going to have damaged onions. They're going to be releasing onion juice. So we wanted to ask the question, if you had the organism in the presence of onion extract, what would happen? And this is a question that's going to come up later. A lot of people believe that onions are antimicrobial. And so this question, well, would onion extract protect the cells during this storage period, or would it actually reduce the populations of the organisms? And in both the cases of Ephesium and Salmonella, onion extract was protective. And so you can see what we're showing here is the reduction. So this is not the population, this is the reduction at 84 days or 12 weeks of storage. And you can see that with Ephesium, there's almost a 2 log difference or 1.5 log difference. The same with Salmonella, a 1.5 to 2 log difference, with less reduction with the onion extract, and very similar results for both stainless steel and polyurethane. Okay, so then we wanted to move on, knowing that there was reasonable survival on these surfaces. We wanted to move to the next question and ask, what are the factors that influence that transfer, onion to surface to onion or onion to onion? And to do this, we chose to use a system where we could control a number of things: the force, the time, and the number of contacts. So this is actually a texture analyzer used by the food industry to measure texture of food, but we were using it in this case to really control each transfer event. And so you can see the piece of equipment with the polyurethane on the probe, and in this case, a square of onion as the recipient. And you can see that we're taking the skin of the onion to enumerate the bacteria that were transferred, and that allowed us to go to a reasonably low limit of detection for these experiments. So we looked at a number of things. We looked at transfer direction. We looked at surface material. There was no difference between polyurethane and stainless steel, so I'm not going to show that data. We looked at the difference between Salmonella and Ephesium, a couple of different inoculum levels. And again, we wanted to look at the impact of onion extract, peptone as a control, and then something we call soil water, which was basically just taking some agricultural obtained soil and mixing it with water, and then using that as the carrier for the microorganisms. I do want to mention, when you talk about transfer rates, how you calculate that. So you're using the number that you have identified on the donor, in this case, 10 to the 7. You enumerate the recipient. In this case, our value is 10 to the 4. You don't convert to the log. So you're looking at 10 to the 4 divided by 10 to the 7 to come up with a transfer percent, a rate of 0.1%. So you're going to be seeing some of those numbers, and you're also going to be seeing some numbers, which is the log percent transfer rate. So just a reminder, 0.1% is -1 log percent transfer rate. So you're going to see some negative numbers in some of these slides. So in this particular slide on the left-hand side here, we're looking at transfer from the onion to the polyurethane and also transfer from polyurethane to the onion. So in the black here, the donor in both cases was not significantly different. So the donor, the onion, or the polyurethane. But in the recipient, there was a significant difference depending on the way you went. So greater number of cells were transferred when you went onion to polyurethane rather than from polyurethane to onion. And this is shown here in an expression of log percent transfer rates for individual samples. Each individual sample is shown by a dot. And you can see that in a lot, even though the control that we had with the texture analyzer, we still get a fair amount of spread in these experiments, but definitely showing here a significant difference, onion to PU, giving a higher transfer rate. Okay, so the second thing that we were looking at, or another thing that we were looking at, is the level of microorganism on the donor surface. And so we looked at a high level, 10 to the 7 per centimeter square in this case. And you can see actually a very striking difference between Salmonella on the right-hand side, which has lower percent transfer. This is a frequency of distribution of transfer rates. So you can see that shift down to the negative numbers or the lower percent transfer rate than the Ephesium, and much more spread out. But interestingly enough, when we went to a lower inoculum level on the donor surface, then we saw a shift for Salmonella. We saw almost no change in Ephesium, a shift to the right, so to a higher level of transfer with Salmonella, now much more closely mirroring the Ephesium. We don't have a good explanation for that, but it is an observation that not necessarily, you might think that as you go lower with populations on the donor surface, you might end up with less transfer, and that didn't happen. I will say we have very similar data when we looked at transfer from onion to onion. As I mentioned, we wanted to look at the effect of onion extract on transfer, and you can see the highly significant difference, an increase in the transfer rate in the presence of onion extract. And we think that was primarily because onion extract is very sticky and viscous. And so you can see the difference on the dried surface between these three carriers, how different they are, with the onion extract being actually shiny and sticky. So you can see the polyurethane coupon after transfer, you can see this stuff has moved. Now, I'm going to say that in a packing house environment, this stuff is likely to get coated with dust and other debris that is coming through the packing house at the same time the onions are, but it is something that I think is of interest. So in summary, we've got this transfer of Ephesium significantly higher with onion to surface than surface to onion, which is interesting. Transfer of Ephesium didn't matter if it was stainless steel or polyurethane. Transfer of Salmonella but non-Ephesium was impacted by inoculum level with transfer rates lower at high compared to moderate inoculum levels. And then transfer was significantly higher in the presence of onion juice. Last slide here, I'm just going to say that we have a paper that was published yesterday focused on peaches, very similar, but we do have one onion experiment in that paper that showed that we actually saw very similar transfer between peaches, nectarines, and onions. So I just wanted to mention that. Okay, so up next is back to Joy and talking about transfer and sanitation.
Great. Thanks, Linda. We also did some work very similar to what Linda presented and very comparable results. So it's always nice to get very similar results in two different labs. But what I'm going to present next is really talking about, are there some potential things we could do to minimize that risk associated with the cross-contamination? To bring us back to the outbreak situation from 2020, we ended up with 3 months worth of packed onions being recalled. In this facility and in the outbreak investigation, there was epidemiological links to red onions, but the facility was handling red, yellow, white, and sweet onions around this same period of time. And through that investigation and the lack of effective cleaning and sanitation, that recall was expanded. And so we had a tremendous amount of food recalled as part of this. So in the recall statement, the statement comes out of the recall needs to be expanded due to concerns that they may have been cross-contaminated via food contact surfaces that were used to process the red onions that were associated with the outbreak. So this is just a cartoon of what in theory is going on, right? So we have, in this case, red onions that are contaminated. They're coming into this packing facility and they're depositing, in this case, salmonella onto these food contact surfaces. And then the next lot of onions that's not contaminated, in this case, a yellow onion, is touching those same surfaces and then picking them up. And so that may present a food safety risk to those consuming the yellow onions. As a reminder, in the produce safety rule, the regulatory expectation related to cleaning and sanitation is that a grower must inspect, maintain, and clean, and when necessary and appropriate, sanitize all food contact surfaces of equipment, tools used in covered activities as frequently as reasonably necessary to protect against contamination of covered produce. And this is intentionally vague, right? Because it needs to be applied across the produce industry. But there's this question of what do you do in an onion facility? That expectation is really guided towards the use of wet cleaning and wet sanitation. And these onion environments, the packing houses, are dry facilities. So there is no water used in these facilities typically throughout the entire growing season. There are some that have changed practices, but generally we see a complete avoidance of water. The use of water is potentially going to increase the risk because there would then be enough moisture for things to grow. So we have this question about, like, how could we potentially reduce the risk of cross-contamination in these types of facilities, ideally without introducing much or any water. So we did do some sampling in an onion packing house just before and after their typical dry cleaning operation. So this is just vacuuming, sweeping, general housekeeping, dry operations. So before, this is an aerobic plate count data set. So before cleaning, we have about 1 million cells per 100 square centimeters on, in this case, we sampled conveyor belts. And then, not all that shocking, after dry cleaning, nothing changed, right? So cleaning activities typically won't reduce microbial load, whether they're wet or dry. And in this case, we just demonstrated that's in fact true. Dry cleaning won't reduce microbial load. Oh, sorry. We know that it's not going to reduce microbial load. So is there something else that we could do to reduce this risk? We're going to ask questions about what is the cross-contamination risk between onion lots? Would dry sanitation significantly reduce that risk? And we're guiding these back on these questions of what would be the pathogen burden in the packing house, because that's going to help guide us into our inoculation level. So we took all the data from the outbreak that we could collect, the outbreaks. We took data from Linda's studies and some of my studies, and everything's pointing to low prevalence of contamination and very rare high contamination potentially if there's culls or rotted onions. In the name of food safety research, we challenged the system to evaluate a worst case scenario, high prevalence and high contamination level. We did this at 3 different scales. We did it at lab scale comparably to Linda's, and we got very similar results. We also did harvest bin scale and commercial scale on conveyor belts, and I'll present all that data today. So I'm not going to present any lab data, but we'll talk about the other ones. And then whether dry sanitizer significantly reduces cross-contamination. And if you're not familiar with that term, these are basically alcohol-based sanitizers. There obviously is water in those mixtures, but because of the alcohol concentration, they're going to evaporate very quickly. So you're not applying really any other water to the system. We used 3 different commercial products. So dry sanitizer A was just 30% ethanol. Dry sanitizer B had 2 different alcohols, ethanol and isopropyl at 30% each, and also contained quats. And then dry sanitizer C had 10% isopropanol, hydrogen peroxide, and quats. So various levels of complexity here. So we did do some lab scale evaluations on a couple of different surfaces. I'm going to show you the high density polyethylene data first. So in this case, we're targeting salmonella. The goal of a sanitation treatment is that it should achieve a 5 log reduction. So that's what we were aiming for, which is the black line. So the closer the bar is to the black line, the better. Hopefully it goes over the black line to be effective. So let's see what we found. So for dry sanitizer C, which had the lowest alcohol concentration, saw very little reduction. So about maybe a 1 log reduction on average. So this one wasn't all that effective. When we jump to 30% alcohol, we do see some improvement in the reduction. And when we get to 60%, we saw a pretty wide spread. So in all of these cases, we had a 30-second total wet contact time, and there was a lot of distribution, but at least some of the samples were getting over 5 logs. So that was promising. We went ahead and evaluated the impact of a drying time, because obviously that's the point of the use of these dry sanitizers in the facilities, is that they are going to dry off rapidly. In this case, when we included the consideration of 15-minute drying time, all of the samples achieved well over 5 log reduction for that sanitizer with the high alcohol content. We found this one to be effective. Alcohol concentration does matter. About 60% seems to be a good point. We also investigated the alcohols by themselves, and that was true for isopropanol, ethanol, and the combination of the two without any other additives. Drying time is an important component of the sanitation process for these dry sanitizers. So we wanted to take the effective sanitizers to harvest bin level. So we wanted to evaluate what would happen when we start to scale up to a commercial environment and evaluate, does this reduce the number of onions that would be positive, whether you sanitize or you didn't do anything. So to do this, we took a harvest bin. We had a plywood harvest bin, and then we also had a plastic harvest bin. And we inoculated not every part of the bin, just a proportion of the bin. In this case, we inoculated 6 squares on the bottom of the bin with about 8 logs of the generic E. coli per square. Then we added 72 clean onions, enough to cover that bottom surface. And then we analyzed the number of onions that were contaminated from that cross-contamination event. This is what it looked like in the bin. So if we look at our transfer frequencies, we looked at both wood bins and plastic bins, and we did that with a sanitizer intervention or with no sanitizer intervention. So we'll do the wood one first. So with no sanitizer intervention, we end up with somewhere between 82 and 97% of those onions being contaminated from touching that surface that was highly inoculated. For plastic, we see somewhere between 47 and 71%. So not as high as the wood, but still the majority of onions were positive with no sanitation treatment prior to adding the clean onions. What about when we add a dry sanitizer? So in the wood bin, we used dry sanitizer B. So that was that product that was effective at lab scale. And we also tested 60% ethanol without any other additives. So in this case, we are seeing a reduction right? So we went down to as low as 19% were positive, as high as 63% were positive. What is a porous surface, right? So sanitizers aren't expected to be super effective on those types of surfaces, but we did see some reduction. What about plastic? So for plastic, the sanitizer worked very well. It's not perfect. Sanitizers are not sterilants, so we wouldn't expect it to eliminate every potential for cross-contamination, but it did a great job. So we got down to, there was 0 cross-contamination for one replicate, and then as high as 6%. This sanitizer is definitely reducing cross-contamination risk in the plastic bins. But we also wanted to see what happened when we used one of those sanitizers that wasn't so effective. So dry sanitizer C was the one with the 10% alcohol and hydrogen peroxide. And in this case, we actually saw an increase in cross-contamination when we used this ineffective sanitizer. So we went from a majority of onions being positive to every single onion being positive in one replicate and 86% being positive in the other case. So what we think is happening here is that sanitizer is not particularly effective, but there's enough liquid that it's moving the contamination around into the bin, and there was probably more surface area that was available to serve as a cross-contamination source. So then we went up to commercial scale. So we partnered with a packing house in the region, and we decided to look at what would happen if you ran a lot of contaminated onions through a series of conveyor belts and then did it again with an uncontaminated batch of onions and what would happen with that transfer. So in this case, you can see we're going to add first our inoculated red onions. We have nicely 2 replicates already in this setup. These onions are going to move through the belts. So as the belt's running, they're going to move from the green source belt of the onions onto the blue belt, which is going to be our test belt. And then they'll move off of that onto another receiving belt. So we've gone through this process. Once all the red onions are off, now our contamination event is complete, and we can swab that surface to understand what the cross-contamination was from the onions to that belt. Then we can load up our uninoculated onions and do the same thing again with or without a sanitizer intervention to try to understand whether the use of a sanitizer would reduce that contamination risk. So we'd run those uninoculated onions, then we would go ahead and, collect each of those onions and test it for E. coli, and we'd also check that surface to see how effective the sanitizer was. Okay. So let's look at what happened. So we did this effectively 4 times. So I'll present that data. We'll talk about just cross-contamination first, and then we'll talk about sanitation after that. To start with, on average, each of those onions was inoculated with 6.5 log CFU per onion. And that resulted in about 3.1 log CFU per centimeter on that blue belt. And this very much matches the transfer rate that Linda presented when she did that lab work. So we're right in the same zone. There was a range. Obviously, this wasn't going to be a uniform distribution of that contamination. So we had somewhere between 2.1 and 4.6 log CFU per centimeter in that first rep. Second, third, fourth reps, quite comparable. And again, here's the ranges. There was more variability in some of these than others, but in general, we got fairly comparable results, in my opinion, for this scale of application. Okay, so what happens when we add our uninoculated onions through this system? And what we found was in the first replicate, we had 5 out of 31 onions were positive for E. coli, and they ranged in contamination levels from 2 to 260 mpn per onion. As we went to the second rep, fairly similar, third rep, and fourth rep. Somewhere between 16% and 40% of the onions were positive, and they're positive at fairly low levels, so somewhere between 2 up to 1,000 cells per onion. So what if we add a sanitizer before this contamination event? Would it reduce these numbers? So In this case, we applied our sanitizer and our contamination level on the belt decreased substantially. It was almost eliminated in all cases. So we had, for one of these, 2 of the reps, we had only 1 out of 10 swabs that still detected E. coli after the sanitizer, and at very low levels. So only 2 CFU, not 2 log CFU, but 2 CFU per 100 centimeters squared and 3 CFU per 100 centimeters squared. So very low levels of contamination. And the other 2 replicates, we didn't detect anything on those 10 swabs. So would this also eliminate cross-contamination to the onions? And the answer is no, it didn't. So there was still enough E. coli on those belts, and how the onion moved on the belt and touched multiple surfaces as it rolled around, it still picked up some surviving E. coli. So in this case, we had 3 out of 32 of the onions were still positive, and they were positive at a rate that was pretty comparable to without the use of the sanitizer. Second replicate, fairly similar, but lower levels on the onions, same thing. So consistently, we had cross-contamination still occurring after the use of the dry sanitizer. Generally, the MPN was in the same range, but the total number of onions that were cross-contaminated was reduced. So, but it didn't eliminate the problem, but it did reduce the problem. So at commercial scale, sanitizer reduced but did not eliminate transfer frequency. And we are working on doing some modeling to actually estimate what the risk reduction would be. So what is our cross-contamination risk between lots? Looking at some of Linda's data, some of our data, don't quote me on these exact numbers, but our data was suggesting about a 0.01 transfer rate, but there was a lot of variability. So it's really hard to estimate that. And then on to the next batch of onions, that would be a 0.001% transfer rate. So we're just decreasing the transfer rate as we move through different contact points. So while the rates of cross-contamination are low, the variability is high. And so this leads to really low predictability, which makes these systems hard to study and also hard to control. Our question about would a dry sanitizer reduce the risk of cross-contamination? It really depends on the dry sanitizer. So if you are using a high alcohol content sanitizer, that will reduce risk in the number of onions that are cross-contaminated. But if you use a low alcohol sanitizer, it could actually increase the risk of cross-contamination due to things spreading around in the facility. Just to point out again, high alcohol sanitizer will reduce but will not eliminate the cross-contamination risk. And again, we're doing some modeling to estimate that. I'm going to kick it back to Linda to talk about cut onions.
Okay, I know we're going a little long, but we're now getting into more the final preparation stage. And I show this picture because these are just some onions in my kitchen, and I encourage you the next time you're cooking with onions to actually have a look at them and try to figure out where microbes might end up in an onion or how they might get there. And I recently got asked a question with salmonella goes through the skin of an onion. And I think really, if you look at the way an onion is structured, that is not a likely way in which it would happen. And as Joy has already shown, and this is again a picture of an onion from my kitchen that had some decay going on, where you could watch that going down the layers of the onion. And you can actually see in this picture between this particular red onion and this yellow onion, how even the structure and the amount of openness that is in that structure might allow for introduction of microorganisms into that system. So anyway, I encourage you to play with your food the next time you're cooking with onions. As was mentioned, this section is going to be on the final preparation. I wanted to point out that in addition to the large outbreaks with whole onions, which obviously ultimately were prepared before consumption, there have been a number of outbreaks that have been linked to fresh cut or restaurant prepared onions. You can see in some cases they were suspected, as in the case of this chicken salad, where onion and celery mix was suspected, or frozen vegetables that included onions. So some of these are not pure, perfect links to onions. I got involved in onions back in this 2008 outbreak that occurred in Canada. It was a restaurant that was using chopped onions in the kitchen, and they had some practices that at the time I was talking to an expert. I was acting as consultant, and the consultant for the other team had an expert that said onions are antimicrobial, and so nothing could survive in it, and so it's just couldn't be our problem. And I really thought, if that's the case, fine, but I don't see any data in the literature. So that was what sent us on this exploration. And we did two things. One, to first look at the onion, the whole onion, because at that time we hadn't done any field trials, we hadn't done any of the work on curing, but we looked at inoculation of the onion, asking the question, onions are stored for long periods of time, both refrigeration and cool temperatures and ambient temperatures. So what would the effect be of this storage? And you can see what our choice was to spot inoculate specific areas of the onion, and then again to remove that onion skin and enumerate on that onion skin. We showed that very little movement went below where we inoculated the onion skin, so that this was an effective way for us to get to, again, a low limit of detection. So I'm showing the data here from two different papers, E. coli, Listeria, and Salmonella. All of them were held at 4 degrees, which is in blue, and at ambient conditions about 23 degrees Celsius in green. And then Salmonella we also held at 13 degrees, which is shown in gold on the right-hand side over here. And so you can see that this is very typical of survival, where the cooler the temperature, the better the survival. So there was a much, much slower decline of each of these three pathogens in refrigerated storage. At ambient conditions, a 5 log reduction was observed at 3 weeks with E. coli 157, at 6 weeks with Listeria, and at 4 weeks with Salmonella. So ambient temperature storage, possibly organisms are dying off over time under ambient conditions on the skin. So we also wanted to ask the question, well, what happens if you chop an onion and you can imagine that if there was contamination on the surface and you are preparing onions in your kitchen, there's lots of opportunity for cross-contamination to the flesh. And as pointed out earlier, there's possibility for microorganisms to be in parts of the flesh as you prepare it. Chopped onions stored at 4 or 23 degrees C for all organisms, and then 10 degrees Celsius for Listeria monocytogenes. So here I'm showing you E. coli in squares and Salmonella in circles at the two different temperatures. Behaved remarkably similarly between the two organisms. As expected, no growth, but then no die-off at 4 degrees in these onions. And at 23 degrees, a lag phase that was in the neighborhood of 8 hours, and then logarithmic growth up to a population of between 10 to the 6 and 10 to the 7. See if you definitely evidence of these organisms growing in chopped onions. One of the things we wanted to do in this experiment, because it was occurring in that restaurant associated with the 2008 outbreak, was this practice of what I call chopping off. You have a container with chopped onions. It's sitting there. You're using them. In this case, it was to put onto hamburgers. And then it's getting low, so you pull it out. You put the new one in, and you put the old onions on top. And so that's what the arrow is. After 12 hours, we moved some of the old onions, if you will, onto new onions, and the new onions were cold at a 1:9 ratio. And you can see what we effectively did was to shorten the lag phase. But interestingly as well, we also shortened or reduced the maximum populations that were observed in this situation. But that idea of topping off will prime the system and get the organisms going into log phase much faster. We looked at listeria at 4 degrees and 10 degrees, and you can see that indeed listeria grows at 10 degrees Celsius. That's not a surprise. It did not grow at 4. And just to point out, the photos were taken at 21 days, not looking terribly edible at 10 degrees at 21 days. But I will say that the time point before this, which I think was 17 days, was not a rejection kind of material. And that's the thing about onions, is that I think they can sit for relatively long periods of time and still be not be rejected by somebody who is trying to decide whether they were going to eat them or not. The last study that I'm going to go through pretty quickly here is one that was inspired by a totally different restaurant that had a different set of outbreaks. But one of the control mechanisms that they said that they were going to convert to in order to reduce risk in the kitchen, and this is where they were preparing onions in the kitchen, they said publicly that they were going to drop the onions, all of the onions that they were going to prepare in the kitchen, into a hot water bath, and that this was going to take care of any problem related to contamination on the onion. And so, again, I thought, we don't have any evidence of this. How can we set this experiment up in the lab? So we started with inoculation at the top, at the equator, and at the root end. We started with boiling water, essentially, for a very short time. And then we also ultimately did a lower temperature, which we thought was probably more manageable long term for varying lengths of time. So essentially, got this inoculated onion, putting it in the water bath, and then pulling it out. So in this particular graph here, I got the log per onion, starting here at about 10 to the 5th. And so this is after 5 seconds. So we're going times 0, 5 seconds, times 0, 5 seconds, 0, 5 seconds for the equator, root, and the top. And you can see that 5 seconds of exposure in all 3 cases, we went down to none detected, and of enrichment of 6 whole onions. Now, when we look at listeria, we got exactly the same result. And when we looked at salmonella, we got the same result for the equator and the top, but we saw not only positives at the root end, but we saw countable numbers of salmonella remaining on the root end. So in our next set of experiments, we focused on salmonella and we focused on inoculation at the root end. So this is where we dropped the temperature of the water to 85, and then we looked at different temperatures. And you can see at 85, you don't get that 5 log reduction or 6 log reduction in 5 seconds, but you get a more gradual decline over this 45 seconds. And we made this observation that we had this one unusual higher recovery, but that particular onion, the root had oriented up rather than down. And so when we took a look at the data, because we recorded whether they went up or down, if you throw an onion into a bath of water, turns out that most of them will freely orient themselves with the root down. But a small percentage, 7% in our case, oriented with the root up naturally. And we decided to explore that a little further. And so we forced the onions to be root up. And as you can see, that doing the same experiment over again with contaminated root area and the root up, we basically got very little reduction of the pathogens during even up to 180 seconds. We did another experiment where we said, okay, what if you put a lid on your water bath or your pot on the stove, whatever. And so we were able to show that this is the untreated here, so no exposure to water, the control. No significant difference with the uncovered treatment. But then when you covered the water bath, you were able to get a mean of 4.9 log reduction with a range from 3.5 to about 5.5 log reduction. And not that people would do this normally, but I was able to show that it might be effective depending on how you chose to control the system. So, in summary, pathogens decline on onion skin, significantly slower reductions under refrigerated storage. Pathogens definitely grow in chopped onions at permissive temperatures, and this topping off reduce the lag phase but decrease maximum populations. And then you can get significant reductions by putting in hot water. We did look at the impact on the quality. There's not a lot of impact, especially with the shorter exposure times. With that, I'm just going to move into a couple of slides. I want to acknowledge all of the people that were involved in working with me on these various projects over the years, co-PIs with me, but also a really great set of staff who helped along the way. And I have a couple of pictures here, but I also have a long list of people to thank. And then, of course, we don't do this work without the cooperation of the onion industry. So we had a lot of great insight from them. And then funding from the FDA as a separate grant, a standalone grant, and through the Western Center, as well as funding through the Center for Produce Safety. And Joy will have a couple things to say about her acknowledgments.
Yeah, so just to echo Linda really quick, the research that I presented was supported by a grant from Center for Produce Safety and from the Washington Specialty Crop Block Grant Program. Phenomenal people to work with, including Linda, obviously, and then Faith, who's one of the panelists here, and then Stuart Wright, he's on the call. So he's our resident onion expert at OSU in Malheur County, and then Tim Waters supported the field studies as the Washington State onion expert. And then Ivana Kovacevic helped with supporting the sanitizer work. Obviously, like Linda said, the industry is such a key part for helping us understand what the considerations are for the industry and practical applications, as well as realistic risks. And then a huge shout out to the Food Safety Squad. That's what we call ourselves. These are some pictures, but like Linda said, there's too many people to think publicly one at a time. But also, one last plug, we are planning a dry sanitizer workshop for the very end of January in Othello, Washington. And hopefully we'll be able to push out some information about that real soon.
Okay, so we did see one question about links to publications, and I want to let you know that this was updated as recently as this morning. It's on my website, ucfoodsafety.uc-davis. We can, I'm sure, send out the link to this. It has a complete bibliography, as complete as I could make it, on research that's been done on the safety of onions, and it includes all of our papers. And all of our papers are open access papers.
Tons of questions and comments. We're definitely not going to get to all of them in 4 minutes, and we are going to be timely. So we will take all of those questions and comments, and we will send out a follow-up document that lists what those answers are from the speakers. And we'll also share those resources.